that we capture everything. So yeah, thanks again for coming along, and Justine will speak for probably, around sort of 30, 40 minutes, and we can make it interactive and have some questions, and we'll have a break before Cameron's talk. So you can stretch your legs, get a drink, and then we'll move on with Cameron. So thanks again. And so Justine, I'll pass over to you if you like, if you're happy to introduce yourself. That would be great.
Yeah, sure. Hi, everybody. I'm Justine Cox. I'm a soil scientist, and my brief is on soil health. So I've been working in the soil health area for 15 years, and my background is in ecology. So I came with an ecologist eye. So I've been working in soil biology for a while, and I wanted to give you an update about what we've been doing, and my colleagues and other researchers and extension people around the country. So if I'll just share my screen, and yeah, just jump in if you've got a question. And let me know if you can see my presentation first slide.
Yes, I can. It's yeah, if you just pop it on presentation view. Yeah, it hasn't. Oh, yeah, there we go. So it looks. So we can see the two slides at once. Yep.
I'll see if I can do that.
Perfect.
Is that better?
Great. Thanks, Justine.
Okay, so I want to talk about like understanding soil biology, managing it, and also monitoring, because there's a lot of interest in all of those aspects of soil biology. So I just want to introduce the soil organisms, and we often sort of group organisms by size because it's very convenient, and it also relates to their function a bit. So on this left-hand side is called the microfauna, and then the very small end of town. So the smallest are the bacteria. So right on the left-hand side. So bacteria, you need a very high-powered microscope to see. And then you've got the fungi, which have a big range of sizes. Some you can see with your eyes, some you might need a microscope to see. And you've got groups, the nematodes. They are these worm-like creatures, and they've got a very large range of sizes too. Protozoa, and you've got this rotifer. And so then you get to the groups of organisms called the mesofauna. And that's about 0.2 millimeters to 10 millimeters. So you can see some of these with your eyes. But if you want it close up, you need a microscope. So all of these organisms run around in the soil and the litter. So things like your mites and your centipedes and collembola and isopods, so slaters. And when you get above 10 millimeters, we call that the macrofauna, and that's the larger thing, beetles and the amphipods. And so because they're bigger, they have a bit of a different role in the soil. So I just wanted to give a quick overview of the organisms and how they're sort of classified. But we're very interested in the function of the organisms in the soil and why they're important. And a lot of it in agriculture comes down to their role in decomposition and associated nutrient cycling and carbon storage and turnover. So these organisms, their primary role is to decompose organic matter in the soil. And in that process, they use organic material and they shred it and use up the carbon and then release the organic forms of nutrients like organic nitrogen, organic phosphorus. And then other organisms slowly convert that to mineral forms where plants then can take that up. And that's what happens in nature, in forests and grasslands. And that's how plants get their nutrients. Also in carbon storage and turnover. So organisms require carbon to function for energy. But they also create different types of carbon molecules. And there's this constant dynamic changing of carbon forms in the soil. Carbon dioxide is released from the soil, and also carbon is inserted into the soil through the roots. So there's very, very dynamic carbon stored in the soil and the roots. And also organisms are really responsible for rearranging soil particles and structure. So they can create pores, holes, and help bind soil particles together. They're involved in suppression of soil-borne diseases. If you've got a very diverse and active group of soil organisms, then they can outcompete or they can produce other molecules to prevent build-up of disease organisms. And there's a couple of different methods and mechanisms where they do this. Also, some of them can transform potentially harmful elements and compounds. So some of the herbicides, fungicides, and insecticides can be converted into less harmful elements. So here I've got a picture of down in the bottom of the fungal threads where you can very clearly see the white threads. So let's take you through the organisms again and specifically about the roles each of the groups have. So the microbes, which are the bacteria and the fungi. So bacteria decompose organic matter. So they sit on plant roots and organic matter and old rotting insects and plant roots, and they decompose what they're attached to and release plant available nutrients. So they do that via enzymes. So they release enzymes, or they have enzymes inside their cells to catalyze that conversion from organic nutrients to inorganic. And while they do this, they also secrete sticky proteins and gums which stick soil particles together and they form aggregates. So they have very important roles in the soil. Fungi, with their larger organisms, they're long strand and thin. They're hyphal threads. That's what the threads are called. They also decompose organic matter and also lignin. So their role is to decompose sort of woody material, and also they can decompose lignin in wood. So they can also penetrate into larger pieces of organic matter and also inside living plant roots. So they wrap their threads around these aggregates, and they also then improve soil structure. They can hold the aggregates together and make them more stable whenever the soil gets wet. And there's a lot of research looking at the mutually beneficial relationships with plants. So there's fungi and plants go together in many, many plant species, and there's so many interactions to discover here. Protozoa then are the single cell organism, but they're mobile, and so they run around in a thin water film, and they eat bacteria, fungi, and organic matter. So these are quite an important group in the food web. And so let's go up to the larger organisms, the microfauna and then the mesofauna. So nematodes. Nematodes are a tiny sort of worm-like organism. You can hardly see them, but they're the most diverse group on the planet. There's 40,000 species that we know of, and there's probably more. 80% of the individual animals on Earth are nematodes, and they are everywhere, and they're mostly beneficial. Although sometimes you hear very bad press about nematodes, that they do cause a lot of diseases, but the majority of them are very essential parts of the soil food web. So nematodes eat a lot of things. Like individual different nematode groups are very specific in what they eat, but there's nematodes that eat bacteria, nematodes that eat fungus, nematodes that just eat protozoans, nematodes that actually eat roots, nematodes that eat other nematodes, so they're carnivorous. And then there's some nematodes that just eat everything. It's garbage guts. And so there's a lot of work being done, look on the community sort of structure of nematodes, which can tell you a lot about the system that they're in. The larger organisms like the springtails, the mites, the slaters, the diplura, amphipods, all the things tiny little things you can see running around, they're they have an important role in shredding leaf litter. And so that makes that increases the area then for microbial decomposition. And then they're predators, so they eat the organisms below them in the hierarchy. So they are predators and regulators then of microbial populations. So microbes just don't grow and grow and grow forever because there'll be predators out there to eat them. So if we want to know where they are, most soil organisms are really active in the top 30 centimetres. And most microbes live in a thin film of water around soil particles or plant roots. So they're actually almost aquatic organisms. If you have a look at this diagram of this sort of plant and root system, if we have a look at the middle sort of picture, that's a root. And so all the organisms are very much associated around the root. And there's like almost a gel or sort of aquatic sort of environment around the root. And that area then is called the rhizosphere. On the right, you've got some fungi sort of around on the outside of the root, are also entering into the cells and proliferating inside of the root. And some of these will be beneficial and some of these might be harmful. So what regulates soil organisms and their activity? Because if we want to know about them and we wanted to see if we can manage them, we need to know what regulates them. It's really vital that you know your soil type, or especially texture, because that's very important in the activity that you should expect in a soil. So from the extremes, like from a very sand will be very different to a clay. And that's because of the size of the particles. So the sands have really large particles compared to clay, which are very small particles. So the spaces in between the particles is like the pore size and then how much water that allows through the soil. And then the penetration resistance, so how hard it is for, say, a root to get through the particles. So texture is really important and will govern a lot of the activity. Moisture is critical. These organisms live in water and live in moisture. So if it's very dry, very, very dry, then you can't expect organisms to be active and abundant. But also within a soil type, then season and climate. So is it the wet season of your year? Is it in the longer term, is it a El Nino, El La Nina or El Nino year? And what parts of Australia are you in? So if you're in hot and wet or cold and wet, that will have a big difference on the activity. And organic matter. So that's food. So it's essentially food, housing, and moisture. So carbon, nitrogen, phosphorus, sulfur, all of the elements in organic matter. So if you don't have any food, then you won't have any organisms. And it's not just carbon. There is a whole complex range of carbon molecules that organisms and diverse organisms will require. And also the rate of replenishment. So if you've got a lot of organic matter and then you've got organisms, then they will slowly convert that organic matter to carbon dioxide and reduce the amounts of carbon in the soil. So is there any replenishment of that carbon? Otherwise, we will lose it. So it could be added through just plant growth, because when plants grow, they add their biomass, they inject a lot of carbon into the soil through their roots. Or if there might even be added organic matter or mulch or manures to the surface and then replenished that way. Oxygen, they're an aerobic organism like we are, so aeration. So most organisms are aerobic, they require oxygen. Some organisms can live in no oxygen, and they are very different to the aerobic organisms, and they use different mechanisms to get their energy and their gas reserves. So you can imagine a very compact soil will have very little oxygen in it, so that might affect the amount of microbes that can survive and thrive in that environment. And we've also got temperature. Temperature is very critical in the organism's activity, and so that's related, say, to season, surface exposure. If you've got a very sort of hot environment and you've got a lot of surface soil exposed, then it's going to be a zone where you have no activity at all. Daily fluctuations, you might have a soil that fluctuates a lot in temperature, and so that might not be conducive to a lot of activity. So that again depends where you are in Australia on temperature. So they're more active when it's warmer up to a point, and we'll get an area where it's too cold, then they won't be very active at all. pH is critical. In general, this is very big generalization. You want your soil between 5 and 9 for organisms. Bacteria sort of like around neutral. Fungi can tolerate acidic environments. But you will have sort of crop requirements which will dictate what pH you're aiming for as well. But you don't want to go too extreme for pH. Disturbance. So there's quite a lot of elements about disturbance, like the frequency. If you have a disturbance and it's done once, it might not have a really big effect. But if you are doing a disturbance to the soil and it's like every month, then that's going to select a particular type of organism that can tolerate that disturbance. And so say if you've got a lot of fungal threads that get exposed month after month, and they're not going to be able to survive in the long term. The magnitude, so if you have a really big disturbance, so like you invert a soil, pick it up and turn it upside down, that's going to have a really big impact on the organisms. And the depth, so if you're just doing something at the surface, that might just affect the surface organisms, and they might be able to be quite happy underneath there. So these are the kind of things to start thinking about. So plant chemicals. So we're talking about those root carbon and other sort of molecules that the plant creates and sends out through the roots to provide food for organisms. And also there are a lot of signaling molecules in that chemistry, and they can sort of regulate and sort of select some organisms. And there's a lot of research looking at how specific those signaling molecules are. Are they just saying, come and get some food, or are they trying to attract a particular type of group of organisms? So I'm just reinforcing that activity is really related to moisture and soil types. So on this graph, there's on the bottom axis is moisture proportion of water holding capacity. So here's 100%. And this is on the vertical axis is the amount of carbon dioxide that is captured, and that's sort of respiration. So that's the organism sort of breathing, which would give you a measure of how active they are. So here's this end on the left is dry and on the right is wet. So a lot of these lines are curved so that they actually are most active in the middle. So not too dry, not too hot, a bit of like Goldilocks, like your best in the middle. And so, but you can see a lot of differences in these lines. So there's different soils will have a different response. So some of these will be sands and some of these will be clays, and they will have a different activity. And microbial activity is also controlled by the amount of available soil carbon. So this graph shows on the x-axis, this horizontal axis, the amount of available organic carbon. And on the y-axis is a index used about activity of microbes. And so with more carbon is more activity. So they really do go to town when there's a lot of food out. So it's like a buffet. You know, if there's lots of food around, everyone's going to be really happy eating all the things they like. So knowing that, is it possible to manage the soil and manage your soil biology? Well, let's have a think. So we know how important moisture is. So what can we do to help them maintain a good moisture? If you can soil cover where possible, and they're really going to depend on your enterprise, whether you're cropping, pasture or horticulture. So if you've got a lot of bare exposed soil, you'll have evaporation at the surface. So if there's a possibility of getting some soil cover, that will help maintain moisture retention. It may be possible to add mulch to the soil and cover the soil completely. You can use cover crops. And if you use organic amendments and incorporate them into the soil, they, depending on which ones, have a potential to hold more moisture in the soil. And you can irrigate. So we're just trying to get a really good, as much moisture held in the soil as possible. And it really comes down to organic matter quality and quantity. So if we can increase plant biomass, that will give us more organic matter in the root zone. And when the roots die and then be converted back to nutrients and those root exudates that are sent into the soil. Cover cropping, you can retain stubble and trash, add mulch, add compost, add manure. And so we're dealing with the quantity here, but quality is also important. So if you have a diverse inputs of organic matter, so they're really sort of long-term, short-term, and then medium-term, and of different types, you'll have a diverse amount of organic matter, and that will directly then encourage a diverse group of organisms to come to that area and thrive. And we'll talk about diversity a bit more. Converting cropping to pasture has been shown to increase organic matter. And here we go, diverse plant species will essentially give you diverse microbe species. Rotation, so if you add a few rotations, that will introduce different plant species, then the different organisms will develop. Stocking rates, you'll have additions and organic matter. And even if you just want to reduce losses, that will maintain a high organic matter. So if you've in an area where you lose a lot of soil in soil erosion, if we reduce those losses, then we're going to improve our organic matter content. Aeration. So it's really tricky and really hard to get air into soil once it's compacted and closed up with little pores. So we've got a few options. Deep-rooted plants have been used to try and get air into an uncompact soil, and that's really hard because if it's compacted, it's really tricky to get roots to down there. So there's some research trying to find some effective deep-rooted plants that will grow. You might want to, if you've got a no-till system, maybe an occasional tillage is something that's will be beneficial. There are some mechanical aerators for this very sort of situation. They might have a corer sort of function, or a punch hole kind of function, or a like a very thin line that they can rip. Organic amendments. There's some research trying to actually look at sort of a ripping and organic matter combo to get organic matter down to depth, which will help with aeration. Also, preventing compaction in the first place is really important. So no heavy equipment on wet clay soil you will then avoid the compaction issue. Temperature. Sometimes we can't do too much about it, but if we can cover the soil surface. We can do that with either plants or in mulch. In some horticulture, you try different coloured plastics to change the temperature underneath. pH, so if it's getting very acid, we might have to use some lime to increase the pH. If it's very alkaline, there's sulfur and other methods. Again, organic amendments are useful to maintain a good pH. If we can choose a less acidifying nitrogen fertilizer, that might help in the long run. But it's also really depends on your crop system and what pH your plants prefer, and what method you have of changing the pH. It's very difficult to change pH. And so some systems you'll get a little bit more bacteria, and some systems you'll get more fungi, depending, say, if it's a tree crop system like macadamias where I work, will be quite a bit of fungi in the system. And again, that disturbance. So depending on your crop system, you might want a minimum or occasional tillage, maybe an option. We could add soil organisms, and there are some examples where this might be useful and essential in some organisms. So rhizobium are inoculants that will be essential for legumes, because these are the bacteria that provide the nitrogen. And so these can be very specific to a particular pasture species, or some of them will be generic. So, but they're essential. Mycorrhiza, which are a fungi, and again, some of these are specific and some of these are general, but mycorrhizae provide a benefit to the plant. They get them more phosphorus, more water, and some mycorrhizae then get food from the plant to the mycorrhizae, so it's a bit of a beneficial relationship. And there are a lot of biological products in the market, and there are so many, and we don't know all about them, so it's a bit hard to sort of judge their efficacy. So you need to know that they have research behind them to show that they are useful. So here's a picture of our nodulation of our pasture legume. So there's an example of a trichoderma product for inoculating a soil for protecting against other diseases. I think that might be onions. And so also then farm chemicals have herbicides and insecticides and fungicides. There are so many chemicals out there. We don't know the effects of each one of those on the biological universe, but I have several colleagues that are working on this, looking at effects of these different, at the moment, herbicides. And I'll give you an example in this talk at the end. Okay, so now we know there's some options to manage them. How do we know if it's having an effect? So we want to know how to monitor. But before we go, I want to monitor my soil biology, we need to ask a question or several questions. So what's your goal? Do we just want to monitor over time and see what's happening? Do we want to use it to diagnose a problem? Is there a problem that you maybe think that soil biology has an input into? Do you want to compare, do some comparisons for some of your sites on farm? Is there, just you want to use the information for a decision support tool? So we really got to get really specific about what we're trying to find with using soil biology. And then what's your time frame? Is it something you need to know this week? Is it talking about this season, this year? Are we just going to monitor continuously? So there's a lot of limitations in monitoring soil biology, isn't there? There are no standard methods. I mean, we've used methods in our research, but other groups in the country will use different methods. And so there's no sort of universal method that, like in Australia, you would get, except for some of the disease assessments. But generally, there are no numbers. There's no guidelines on this is low, this is high, and this is good. And often there's no direct relationship with plant productivity. So you might have the plants going gangbusters, but the biological habitat is not that interesting or diverse. So there are a lot of limitations, and we are exploring and expanding on our knowledge in soil biology. And the CRC projects are doing that in spades. Here I just wanted to add that soil biology is important for a lot of soil health monitoring that growers and grower groups have really sort of led the way in. And here's the Northern River Soil Health Card. I was involved in developing that with a whole range of colleagues and the soil care group. And so they wanted some ways of monitoring things that were not just the chemistry lab results. And then the Victorian Soil Health Guide has a component of soil biology in there. So I just wanted to throw those up as useful things to go and have a look at. But this is the guts of what you wanted to know, is how do you test soil biology? And I've separated into in the field and in the lab. In the field, we can count things that we can see. We need to take good samples or good methods, but we can count earthworms in a known volume. We can count mites and ants and those Calimbula and dung beetles. And we also can count the percent nodulation of legumes. So like, you know, how many nodules on this plant are red, and we know that they're active. So that's one way we can test. And we want to know, well, what about the activity? So we really don't need to know who's there, but is there activity and the function of the soil happening? And there's a lot, and we're really focusing on decomposition because that's our main action that we want to sort of see is happening. And there are a couple of different ways you can do that in the field. One is using cotton strips, which is just unbleached calico placed in the soil. I'll show you a bit more. And then the organisms decompose that, and you can take that out after time and have a look. Which is similar to Soil Your Undies program, which is getting cotton men's underwear and digging that into the soil and digging that out and taking a picture. Litter bags, which you place the litter in the soil in a bag with a mesh screen so that organisms can come in and out. And then there's the tea bag index, which uses a tea bag. There's very specific tea bag, two tea bags. One's rooibos and one's like a normal black tea. And then you see how much of the tea leaves are decomposed. And activity, then the product of decomposition is respiration, so the release of carbon dioxide. And there's a commercial test that measures that carbon dioxide, but really easily. It just turns a gel pad or a different color, and then you can determine how much carbon dioxide is coming out. Indirectly, we can look at the percent ground cover, because that will have an indirect component to the soil biology. We can have a look at root volume and distribution. That's a bit tricky. You need to get a really big spade if you've got a big plant. And there's a physical Emerson dispersion test, which actually is related to soil biological activity. If you place an aggregate in water and you see if it disperses, this is slight and then moderate and severe. If it just collapses, then you know that there's no glues or those from that bacteria and fungi holding those aggregates together. So you've got a very good, high active, really biologically active soil. Then you might often see nothing happen when you add water. It just holds its shape and its structure.
Justine, would a slaking test also give the similar?
It's the same, yeah. It's a slaking test.
So yeah. So you might not get dispersion, but you might get slaking, and so even slaking is an indicator that it's not really being held together by anything.
Yeah. Yep, because the biology is related to the physical structure of the soil. So here's a few pictures to show what I was talking about. So the top left one is calico strips. And you insert that vertically into the soil with some coming out so that you get the soil ground level and above and below. and then that will decompose over time, and you'll get losses in the calico. You can pull that out and then have a look. You can either just use it as a visual tool to show the amount of decomposition. Researchers actually then test the calico to see the force required to break that strip, and then you get a number for that, and then we can get then some quantitative data. The other way I've actually used this is actually take a picture, and then there's a free software that just so this will have lots of holes in it, and if you take a picture on something contrasting, it then can calculate the area. So if you have a lot of decomposition, a lot of area lost, it will calculate that. And so underneath that is soil undies. So you get these white cotton undies, and you also put them in the soil, and you come out and they're all degraded. And so there's a global campaign around the world where farmers are encouraged to soil your undies and take a picture and upload it to the global website. And Oliver Knox and UNE at Armidale is really coordinating a lot of this in Australia. The top right is that Solvita respiration test, and I use that a bit. It is you put soil in these jars that they give you, and you put this paddle in there, and you leave it a certain amount of time, and this purple here, will turn a different colour, and that will be related to how much carbon dioxide is evolved from that time. And that's really simple, and you get a 3, and that will tell you what that means. It doesn't in itself, it doesn't actually mean a lot, but if you're comparing things, you can then compare things very similarly. And these are the 2 tea bags for the tea bag index. So they're a Lipton Rooibos and a green tea. So unfortunately, you have to get them from the Netherlands, but I've got multiple packs. So it's if people are interested, I can let them know more about that. That's backed up from researchers. And that's also a global repository of data and engagement. So in the lab, we can count numbers or presence of organisms, and we can do that in bacteria and fungi. We can do that with plate counts, which grow them on agar, or looking down the microscope, but more popular and more encompassing is DNA and other molecular methods. You get all of the DNA of everything in the soil. You can look at nematodes under the microscope or by molecular methods. The percent root infection of mycorrhizae, so how much of the root is infected with mycorrhizae. Microbial biomass carbon, so how much of the carbon in the soil is due to microbes. So that's a good a tool to work out the percentage of the carbon is microbial. And there's quite a number of range of things to look at activity and function. So looking at the enzymes themselves, which actually is the conversion from organic to inorganic. And they can be very specific, like a single enzyme transformation, or general. So if we look at beta glucosidase, it just looks at the glucose conversion. But if you look, there's this test called fluorescein diacetate, looks at all sort of general decomposition. We can do decomposition rate in the lab as well. We can get soil samples and put cotton strip tests in the soil in lab. And we can also measure respiration in the lab. Potentially mineralizable nitrogen. So bring some soil back and estimate how much of the nitrogen will be mineralized under controlled conditions. Then we can look at diversity. There's a lot of testing going on looking at diversity of microbes, and that's a lot of the DNA tests. using different parts of the genetic information of all the organisms in a soil sample. We can look at the community analysis, so especially nematodes. If we know what proportion of the nematodes are, if they're all of the nematodes in the sample were like bacterial feeders, then we can tell us something about the soil in that we'll have a lot of bacteria, or so the composition of the nematodes will tell us a lot about the habitat. Microbial function, and so looking at the carbon use. So this is MicroResp, which is a tool that our colleagues use here to estimate the diverse use of different carbon molecules. And so we can separate all of the different molecules and see the diverse sort of a pattern. And then there's the genetic function. So going right just to the genes and looking at whether we have variation in all the genes responsible for all of these conversions, and that's called metagenomics. There are other indirect ways to look at soil biology, like looking at the carbon fraction. So there's the total organic carbon, there's labile carbon, there's the short, medium and long-term sort of stable carbon. We can look at all of those. We can look at disease organisms, so the predictor-B test. And we can grow plants in what's on this picture on the bottom of the rhizo box and look at root studies and see what's happening in a controlled environment. So just quickly, an example of some research that colleagues are doing. This is Mick Rose and Terry Rose and Lucas Van Zweet. They compared the effects of glyphosate and Roundup. So Roundup is just glyphosate and a surfactant. And so they use substrate-induced respiration, so that microresp test, to look at respiration and enzyme activities that are responsible for the carbon, nitrogen, sulfur, and phosphorus cycling. And they looked at 3 soils in the lab over 27 days. And what they found at label rates, there wasn't any really effect on either respiration or the enzymes. So no evidence there of action. But at high rates, the sandy soil was actually affected compared to the other soil types, which will be more of a clay loam or not sandy. They found that enzymes were not as sensitive as respiration. So that's helping us determine what tests are going to be more useful. So if they're sensitive, we might be able to pick up effects more easily. So that microresp test is actually coming to be very useful. And they found that Roundup affected soil function more than glyphosate. So the Roundup has that surfactant, and so that component was actually more detrimental than the glyphosate. And one last one is effect of herbicides on soil function. So Mick Rose compared 7 herbicides and 1 fungicide. They're all of the chemicals there. I'm not going to try and say all of those. And they measured 8 enzymes, the mineral nitrogen, to see what effect it would have on 5 soils at the recommended rate, and then 5 times the rate. And they found that all herbicides at recommended rate had little effect except atrazine. Atrazine did affect the soil function. So at 5 times the rate of the metal sulfuron, or the 2,4-D, they did find that caused alterations to the mineral nitrogen levels. So if those rates were accidentally put on higher, or they accumulated over time, then there might be some changes that we can pick up. And again, nitrogen transformations were more sensitive than enzyme assays. So we're honing the different tests, we'll be able to work out which ones are more sensitive. So just to summarise, so soil biology testing is complex, and there's no optimum or min-max numbers for each crop, climate, soil type, because imagine Australia is so diverse. but they have value in comparisons or monitoring over time or answering specific questions. So build your own monitoring schedule and then you'll build your own data set. And it's likely that a suite of mixed indicators will emerge as useful for management decisions. It might be a combination of something which tells you about numbers, something that tells you a bit about activity, and something tells you a bit about diversity. So keep involved, keep on the cutting edge, and keep getting dirty. And I'll just end you with some further reading. There's some amazing things going on. Thanks for listening.
That's great. Thanks, Justine. We've got a few questions in the chat box. So Helen's asked about, and I think this is a question that we're all wondering about, is there some sort of form of guidance when considering biological products?
You have to do your own research on your own property. Unfortunately, there's so little known and we don't know how it's going to impact everyone's particular crop or soil or climate. So essentially, if you're really interested and you think that there's valid reasons for it, then you set up your own trial.
Yeah. And
I might just jump in there. Sorry, Justine.
Yeah,
a colleague of mine, Anders Claessens, is a mycologist, and we're currently doing a project with Sumitomo, who are a large chemical company, but they're trying to branch into microbial inoculants, and they're doing some work looking at avascular mycorrhizal fungi as an inoculant and how they affect growth rates on various crops in Australia. So we're doing that in partnership with the Northern Growers Alliance. And what we've found so far is that there's a whole bunch of different fungi that you find in the soil. And so if you inoculate your soils with those fungi, people are going, that's great, look, we found them in the soil, there they are. But not all of those end up in the roots where they're actually needed. So just because you're inoculating your soils and you're increasing that composition doesn't necessarily mean that that's relating to the function or what's needed in that plant. So microbial inoculants are an incredibly new space. And as Justine said, unfortunately, there's no blanket guideline yet. But at the time, the research is currently being undertaken. So.
Great, thanks, Cameron. And some advice I've been given was, depending on what the product is or the company, I'd be asking them for some of their research results or trial results, and asking specifically, if I use this product, what outcomes can I expect, and then building a trial around that to prove or disprove those outcomes possibly. So I know it's a tricky one, Helen, because and everyone out, those that are doing trial work with farmer groups, a lot of people knocking on your doors saying, we've got this product, do you want to try it out? And I think it's really important to get from them exactly what it is that their expectations would be for that product, and whether it aligns with what you guys want to test with the groups as well. So
that reminds me that there's a webinar that our biometrician did. I can give you a link, which helps you run through, if you're setting up a trial, what to do.
Yeah, that'd be
not specifically about biological products, but any trial. So if you're interested, I'll find that.
That'd be awesome. Thanks, Justine. And Helen's also said we don't really get earthworms, which I'd be interested to know after this rain, Helen, if you did go out and dig them up, because I think. And Justin, I might be wrong, but I find that the earthworms will bury themselves, sort of looking for moisture, and then they tend to come to the top, when it rains. Would that be right?
Some species live in particular parts of the soil profile, and some are very active up and down. Some live really low and don't go up, but they definitely monitor the moisture levels.
So she's saying, but we do have termites. Are they a good indicator of soil?
They are, they are too. You use what's in your environment, what sort of yeah, they're a component of the ecosystem, so they would be good to include. I know a lot of the Northern Territory sort of mining rehab work, they use ants because that's what is their dominant sort of group of organisms that tells them what's going on.
Like the canary in the coal mine.
Yeah.
And Ellen was also wondering about what the high rate was from the glyphosate and Roundup work. Was it like a double rate or
5 times, I think.
5 times, okay.
Oops. Let me go back, see if I can.
Previous one, Justine. The first. Oh, yeah.
So 0 to 75 milligrams per kilogram.
Oh, yeah. Okay. So, yes, high. I've just put high rates. I have to go back to the actual. Yeah, it'd probably be 79 milligrams a kilogram.
Great. If anyone else has got any questions, either pop them in the chat or feel free to just unmute yourself and ask away. Something that came up with a group that I was talking to about rhizobia and the pH, which you sort of touched on, Justine, but we were talking about the fact that different rhizobia have different tolerances for different pH, which might be different to the actual crop tolerance for pH. So some crops, lupins or peas or fava beans have different pH tolerances, but their rhizobia might have a bit of a different pH tolerance. So understanding that, I think the GRDC have put out some work on that as well. But I guess it just highlights the importance of digging up your plants and checking that active nodulation and that sort of thing. And you spoke briefly about fertilizers or the fact that that the microbial bacteria or the biological population needs some nutrients, EKS, whatever, to live and survive. And I guess there's a lot of interest in what effect if you are feeding the soil fertilizers, synthetic fertilizers, what effect are they having on your microbial or biological population? And a bit like the assumption about herbicides perhaps bombing out our population, is there a risk that the fertilizers are doing the same thing, that we're putting in some instances 200 or 300 kilos of urea on a canola paddock to punch out some high yields and grazing value and those sort of things? What are we doing to our nitrogen fixing bacteria, or not just rhizobia but other bacteria that might be in the soil that's cycling nitrogen and mineralizing nitrogen? Are we bombing them out and making them lazy, or?
Potentially, if you've got a high nitrogen environment all the time, then you're going to select organisms that either can use that, and you'll deselect organisms that would have provided that function. And you sometimes find that in blueberries. Mycorrhizal function is not even useful and not present because the fertigation system delivers nutrients right to the root system. So you might find the same thing in the bacteria that are responsible for converting nitrogen to mineral form. They're just not useful, or they don't required by the plant, so they're not selected. So you'll just get organisms that will respond to that environment.
Okay.
The plant doesn't mind how it gets its nitrogen. If you're feeding it buffet all the time, it will just go yum.
Yeah. Great.
Felicity, I had a question. Justine, hi. At a couple of the farmer days we've had recently, there was a lot of conversation around the effect of no-till and it almost being a victim of its own success in terms of organic matter in the soil. And so we're seeing that that organic matter breakdown is being quite efficient, and that there's not a lot left remaining, and that a lot of the sampling shows that, you know, a lot of it's being used up. I mean, you've got standing residue, but within the soil, there's not a lot that actually makes it from the surface into the soil. And so the question was, like, what role do those microorganisms have in building carbon, you know, through decomposition themselves? What do they add to soil carbon? I think that there's been, you know, there's a lot of conversation about how we build soil carbon as opposed to the carbon flows. So yeah, it's something that tricked a few people and I couldn't answer it. Just wondering if you've got any idea on how to answer that.
It is very complex. Yeah, because in theory, we would imagine with a lot of carbon inputs and with the diverse organisms, you would be then creating some of the long-term stable carbon, the humus molecules, and you'd hopefully, but it may be just in such small proportions that sampling can't pick that up, and it's in zones that are smaller than a sampling core. So it may be happening, but it's at such a micro scale that we can't measure it. But it still might be happening, and it's a slow process. Yeah, you get something called carbon priming too, when you get extra carbon being deposited in the soil. Yeah, it's very tricky because you might get some dissolved carbon depending on soil type that's going to go through the profile in the solution and the water through the soil. But if you've got a really good active bacterial fungus sort of system, you're going to get a lot of carbon turnover and losses to carbon dioxide. So, yeah, it is. I can understand the dilemmas because there's a lot of things happening under that no-till system. But yeah, I'm not really a good one to answer that one. But my carbon colleagues maybe, like you have to ask Suze Orgel.
Thanks. Yeah.
Yeah, it's a biological system, isn't it? So, you know, the microbes and the biology are going to be using that carbon, but they're also carbon. So as they die and break down, they're actually adding to the carbon pool as well. So, but it's always cycling in a dynamic living system.
I'm going to touch a little bit on the mechanism of how it works in my talk as well. So.
Alrighty, well, if no one else is going to jump in, we might just have an 8-minute break. We'll come back at 11:15 Australian Eastern Daylight Savings Time. Emma, I think that will be 10:15 for you. So thank you very much, Justine. I really appreciate your input, and you're most welcome to hang around and join, come back at 11:15. And we'll pick up with Cameron then. So I'll just start recording again. So thanks very much. Thanks again, Justine, and thanks, Cameron, for making yourself available. We're really interested in this group hearing about the work that you're doing with your PhD in the cropping zone. So most of these guys online will be working in cropping areas. So thanks very much, and I'll hand over to you and let you introduce yourself if you like.
Cool. Hi everyone. So I'm Cameron. I'm the PhD student with Southern Cross University, and my project is funded out of the Soil CRC. I might share my screen. Where are we? Share that. Is that working?
Sorry, yes, that's perfect. Thanks.
Yeah. So I'm supervised by Terry Rose, Mick Rose, and Lucas Van Sweeten, all who are involved with the Soil CRC as well. So my project is looking at understanding the mechanisms of soil microbial function and the role that they have in cropping systems. So Justine gave a fantastic overview of how soil biology is very important in our cropping systems and in our soils. And so what I'd like to do is sort of go in a bit of a deep dive in how those processes or mechanisms actually work. And I'm sorry if I lose you along the way. It just mainly because that doesn't always need to know that kind of information for cropping system roles. However, the message we're trying to get across is that they are very complex systems, and so there's not always a one answer or one solution to these problems. So what we have, there's many influences on the soil biology and the composition of microbes that exist in the soil. And so with tillage, tends to get a very bad rap. However, conventional tillage systems, what tends to happen is that turnover of soil, what they find is immediately after tillage, there's a burst of microbial activity because there's carbon pools and nutrients that microbes might not have had access to due to either compaction or fungal hyphae networks might not have reached those pools yet. And so that turning over of the soil, those microbes now have access to a whole bunch of nutrients and carbon. And so the activity, the microbial activity just bursts. And then we see this new change in composition. That being said, over time, there would have been a community of microbes that have developed at some kind of equilibrium. And that tillage system would have disrupted that equilibrium. And so therefore the microbes will have to adjust. There is conservation tillage, which is only disturbing a small amount rather than sort of a large proportion of the soil. And therefore the idea of this is to still turn over some soil, but not completely destroy fungal hyphae networks or fungal enmeshments and root systems that might have stayed in the soil. And then obviously we're talking about our no-till systems. And these have other issues such as soil compaction. And so therefore, as Justine said, a lot of the soil microbes are aerobic and they need oxygen. And over time, if that soil compacts down, it's very hard to get oxygen in there. So those microbes over time do need that soil to be turned over. Crop management is a huge impact on soil biology and microbial composition or diversity. So rotating crops, obviously, as Justine said, if you increase your diversity of your different species, you're going to get different microbes living in your soils. Cover cropping is fantastic in terms of, well, the idea, the theory behind it is to put more carbon into your soils. However, Terry's currently running a project at the moment that shows that in Australian systems, the cover cropping over summer is actually taking out more carbon to grow the crops, and therefore we're left with a carbon deficit by the time they try and grow your cash crop in winter. Intercropping, companion cropping, again, is great. You've got an increased root system helping the microbes function. Obviously, monocropping still exists and it's still part of the system. And although there are a number of negatives, one being diseases and nutrient deficits, the benefit of monocropping would be that you're developing a system where the microbes know what's coming. They can anticipate some sort of environment, and then they can develop an environment for themselves and develop like a feedback loop with that system. So although, yes, the detrimental effect of monocropping far outweighs its benefits, there are some benefits to it. And then precision sowing. So there's systems where roots are competing in the same nutrient pool or carbon pool in the soil. So precision sowing is allowed to develop optimal spaces between plants so that they're not competing for those same pools in the soil. And then, as Justine said, there's a lot of chemicals and inoculants that have detrimental effects to soil microbial biology. But the inoculant side of things, I'll explain later in my talk how they don't necessarily mean that you're going to get an increase in function. So soil, and I do apologise, some of these concepts Justine's already covered, and it's just the nature of the beast, I guess. But I will skip over some of the slides that Justine's already covered. So some key soil functions that microbes are involved in. You're looking at the bioregulation of plant hormones and diseases, some soil organic matter and carbon turnover, nutrient cycling, soil structure, and soil water availability. So I'll take you in a bit of a deep dive into how those mechanisms come about and the microbes that are involved, and hopefully we can show you that a diverse microbial profile is very beneficial to our soils. So not all bacteria and fungi are beneficial. And definitely if you increase your inputs into your soil and promote microbial growth, you are going to get an increased chance or risk of pathogenic bacteria and fungi. But that being said, generally the arrival of pathogenic bacteria and fungi also means the arrival of competitive plant growth promoting rhizobacteria that compete with those pathogenic bacteria for nutrients. More often than not, unless you skew the environment to the pathogenic bacteria, PGR, PR are very good at their job. They're very efficient at getting those nutrients in the soils. And so there's also, we're looking at AMF, or arbuscular mycorrhizal fungi, and they have an ability to regulate plant phytohormone response. And they do this by attaching themselves to the roots and sending signals through the plant, and the plant then responds with its own signaling. And we get these signal molecules being released into the root system, and therefore the root develops a form of systemic resistance against the pathogenic bacteria and fungi. Another method that microbes utilize is through actinomycetes. Now, actinomycetes are bacteria that are shaped like fungi. So they kind of work as a link between the two microbes, and a common species, phyla of these are streptomyces. Now they're involved in sort of disease suppression in a number of ways. The first is pathogenic antagonism. So this is, it basically disrupt the ability for pathogenic fungi and bacteria to reproduce, so to speak. But another way is they produce antibiotics. So streptomyces have been known to suppress bacteria by producing their own natural antibiotics. When we're looking at soil organic matter and carbon turnover, so hopefully this will answer some of Emma's questions. Soil organic matter decomposition is probably one of the biggest utilizations of soil microbes that we're looking at the moment. And it's because it has their ability to decompose soil organic matter is varied at many different stages and levels. So we get different, as you can see, we have different types of bacteria and different species of fungi as well. So they act in different, utilize different mechanisms, sorry, I should say. So whether they're acting directly on soil organic matter that's come from leaf litter, or it's root exudates that have released by the plant, and these carbons have different compositions. And so it requires different microbes to act on them. However, some microbes have an ability to decompose a wide variety of carbon molecules. And so we have fungi that produce enzymes that will then decompose the organic matter even further. And it's generally up to the actinomycetes to really hit home the more complex decomposition, but also to further decompose the soil organic matter. So as Justine was saying, there's different levels or different types of decomposition. So this type of carbon that you're getting in your soil isn't always going to stay that way. It's forever changing. And this ends up with our soil organic carbon pool. Now, the reason why it doesn't always result in a large amount of stored carbon in the soil is microbes utilize this carbon for their own function. So whilst they are responsible for, some microbes are responsible for that carbon pool, other microbes compete with the plants to take it out of the soil for their own function. So they utilize that carbon. And also some of it's lost due to leaching. So there is the carbon pool doesn't always necessarily physically stay in the same spot as well. So sometimes it's pushed out through a number of environmental factors. This whole process also, so the carbon cycle, if you will, soil microbes, especially aerobic soil microbes, respire like us, and they produce carbon dioxide into the atmosphere, which then the plant then takes back up and excretes as root exudates. So that's your full carbon cycle going on. Unfortunately, soil microbial respiration is a large factor of the degradation of soil organic carbon. So the utilization of that soil carbon for their function does result in an upregulation of soil carbon dioxide in the atmosphere. There's a lot of talk about nitrogen before, and hopefully this will answer some of those questions as well. So some of that decomposition of soil organic matter will end up as organic nitrogen. So there's different bacteria and fungal decomposers that specifically create organic nitrogen in the soil. And then other microbes then act, particularly bacteria, will act on that organic nitrogen through a process called mineralization. And they turn that organic nitrogen into a plant utilization form, which is ammonium. So plants can uptake nitrogen in the form of ammonium. However, just like the mobilization or mineralization of that organic nitrogen, again, microbes utilize nitrogen for their own functions and their own proteins, and therefore that nitrogen is then immobilized back into an organic form. So once the plant has uptaken that nitrogen, it doesn't always finish off the pool of nitrogen sitting in the soil. So there's another process called nitrification, and this has two steps. So Nitrous amanith is a bacterial genus that nitrifies bacteria into nitrite. Some of this nitrogen is also lost to leaching in the soil. Again, another reason why some nitrogen deficient soils exist. Further nitrification can occur and will result in nitrates. From this, though, plants can uptake some nitrogen through nitrates through assimilation. There's also another process that proteobacteria are involved in that is called dissimilatory nitrate reduction to ammonium, and they turn this nitrate back into ammonium, and then the plant can take it up that way. Further denitrification results in atmospheric nitrogen. So kind of like the carbon cycle ending up with carbon dioxide in the atmosphere, denitrificans is a bacterial species that produces this atmospheric nitrogen. Rhizobia can then fixate that nitrogen directly to the roots. So rhizobia are your root nodules, and they convert nitrogen in the atmosphere to plant available nitrogen. However, there's also free living fixators known as Azobacter, and they don't necessarily live on the roots like rhizobia, but they can still fixate atmospheric nitrogen. So microbes are also incredibly useful in our soil structure. So once after the decomposition of all of these organic matters, whether it's through root exudates or leaf litter or whatever, that decomposition can result in a whole bunch of different carbon molecules. Some of these are in the form of glues like glomalin, which is a common fungal glue, and they result in the binding and soil aggregates. So these, it's all interconnected, and there's many different types of microbes that are involved just in this one process. Fungi also employ a sort of a different method called fungal enmeshment. So these hyphae networks are all interconnected. However, the closer to the root, you'll find the greater the density of this enmeshment, and they literally change the shape of the soil around them to suit, and they're very efficient at getting nutrients out of the soil. So they'll actually move the soil around so that they'll create these highways between plants, but also between the plant root and these carbon pools. Microbes are also involved in soil water availability. So as Justine was saying, soil microbes are incredibly influenced by soil water, but they also are responsible for the plant's ability to access water. So through that soil aggregation and soil structure, they allow for plant roots to more readily and easily find soil water, and that's because the microbes around the root tips are also looking for that water. That increase in soil water also increases microbial activity. So as Justine was saying, a drier soil tends to have a lower amount of activity. However, there are different species that rely on different amounts of water. And what we're finding in Australian soils, due to how dry they are, we're finding a whole suite of soil microbes that are now very efficient at utilising very small amounts of soil moisture. Soil microbial biomass is a fantastic indicator of how much water is actually in your soil. So small, not just small amounts of one particular species as well, but also a drop in diversity or drop in composition can mean that, so a lack of soil water means only those particular microbes can utilize low amounts of water, and that's why we're not finding any other types of soil microbes. There are other microbes, as Justine pointed out. So protozoa are particularly, their main role is to feed on the bacteria and the fungi that have taken up carbon and nitrogen in that organic form. Once the protozoa have finished sort of their eating, if you will, of the soil microbes, they then release that carbon and nitrogen back into the soil. So that locked up, immobilized nitrogen and carbon and other nutrients that the fungi and bacteria have utilized for their own function is now back and available in the soil. They also eat pathogenic bacteria and fungi. So it's another form of disease suppression. Algae are incredibly important. They are the pioneers of soil. They are autotrophic, so they produce their own nutrients, and they do this through either light or inorganic chemical reactions in the soil. And they are a great source of carbon and nitrogen for bacteria and fungi. And then you've got your nematodes, which Justine has touched on quite a bit. So I won't really go into too much detail other than they are regulators of bacterial populations. So if bacteria or fungi get too big for their britches, nematodes start feeding on them and bring them right back down to sort of like a happy equilibrium. Justine also has already showed the spatial influence of microbes, so I won't go into too much detail other than the different zones and the different functions that occur in those zones. So your bulk soil is obviously looking at just your carbon turnovers. Your rhizosphere functions are much to do with your nutrient uptakes with your plant roots. Rhizoplanes are where they're kind of the gatekeepers of the root interface. So your root exudate, the release and uptake, it really comes down to the microbes that exist on the rhizoplane. And then your endosphere is where your AMF mycorrhizal fungi exist, which are the cells inside the root. And that's where a lot of your plant hormone and signaling occurs. Another major influence on soil microbes is time. And soil microecologists are trying to marry up a lot of the terminology between sort of what we exist as like above ground ecology and below ground ecology. So they're trying to use the same terminology so that people have a better understanding of what's actually going on. So in ecosystems, you have R strategists and K strategists, and they existed on different sort of time periods. So before this terminology was brought in, soil microbes were looked at as oligotrophs or copiatrophs, which is basically the amount of nutrients that exist. So copiatrophs need large amounts of nutrients to function. Oligotrophs can function on small amounts of nutrients. Now they're switching to the other terminology, which is your R strategists and your K strategists. So your R strategists are your, if you put in you have a tillage system, or you have you've just sown and you've got germination and new crops in your soil, your R strategists are your early microbes. So they're the ones that they're very efficient at growing. They tend to grow in larger numbers, so more of them, but they aren't great at competing with other microbes. So the way they deal with this is they just produce lots of themselves to access that nutrients. They don't take up a lot of energy, so they don't use a lot of nutrients for their own function. Whereas K strategists occur much later, and there tends to be less of them, but they are very efficient at utilizing the nutrients around them. So they're very good at competing for those nutrients, and they tend to come out on top if it was a battle between the two. However, they do require large amounts of nutrients for their resources. As Justine pointed out, there's been advances in the technologies that we utilize to study soil microbes. The biggest one was going from culture-dependent to independent technologies, which means before we used to have to extract microbes from the soils, grow them out on media, and then test them. We now have technologies that we can test directly in the soil. Genome sequencing was huge in terms of understanding soil microbial compositions and starting dichotomy trees of evolution of microbes. However, the problem with that is the technology is so efficient at detecting genome sequences in microbes that the computational power or the stats required to understand how they affect the environment that they're in isn't up to scratch yet. So most diversity papers that I've read, they're using some kind of supercomputer, and the software programs that they're running so far advanced that only really biometricians understand what's going on. So it is slowly catching up, but the rate in which we're getting new information on these soil microbes genetically is far outreaching the ability to understand the stats and the mathematics behind what's actually happening. And Justine already showed that there are many different bioassays that we can use for real-time microbial respiration, which is basically a measure of their function. So if microbes are doing their job, they're generally, as most microbes are aerobic, they're going to be producing CO2. So this is just a little slide to demonstrate. This is a study that was done to show that within major cash crop types, there can either be like many different types of the same microbe, or there can be the same microbe can be found in many different crop types. So it's to show that although soil microbes can be specific and selective, they are found right across the world in many different crops, and this is the same species. And the latest research and what I'm trying to do is show that might, just because they are there doesn't necessarily mean that that's the function that they're doing. And this was basically a whole change. So soil microbiologists changed their way of thinking in terms of the presence of a microbe relating to its function. And this is where we've now changed our approach to soil microbial research. So what used to happen was if there was an upregulation or an increase in a particular microbial species, then that particular microbial function would obviously increase, and therefore that would contribute to a larger soil function, whether it be nutrient cycling or disease suppression or soil aggregation. What's actually happening is it's much more complex. And so many different species are working together on the same microbial function, which might be, denitrification, or it could be complex soil organic matter decomposition. So those microbial functions either utilize many different species of microbes or an overarching soil function, whether it be an increase in sort of pH or temperature, might require many different types of soil microbes. Those soil microbes could also be acting on many different functions. So just to say, if you increase population or species number one, does not necessarily mean that soil function number one would also increase, as population or species number two might outcompete that sudden influx. And this is the missing link of information when we're talking about microbial inoculants, in that soil microbes exist, they like to exist in some kind of happy medium or equilibrium. However, because of the dynamic nature of their environment and processes such as lateral gene transfer, where they can actually share genetic information sideways without having to reproduce, bacteria can actually pass on genetic information to other bacteria without having to reproduce themselves, means that they learn in real time. And so if there's a sudden increase in temperature or pH, most bacteria wouldn't be able to handle it, but there might be a few species that can. And so by increasing your diversity in your soil profile, you're increasing your resilience, I guess, of your soils, and therefore possibly having a better shot at whatever function that you're targeting. So just a quick overview of experiments that I'm running for my PhD. I'm basically going to inoculate some soil types, and then what I'm going to do is, before that, I'll sterilize the soil with gamma irradiation, and that should cease all microbial activity happening in those soils. And then the inoculation step, where I'm going to take from 3 different soil types, and they're all going to do a round robin sort of thing. And I'm going to see if the community composition changes based on the soil type or that they've been put in, and whether they, how far away from their original soil source composition they go. And then whilst that's happening, I'm going to use micro respiration and enzymatic testing to see whether that compositional change in the microbial community actually has an effect on their function. At about 28 days, which is sort of the changeover period between your R strategist and your K strategist, I'm going to send off some of those samples to AGRF, which is the Australian Genomics Research Facility, and I'm going to get a full community workup done to see what microbes are actually there, and look at how much of that particular, so what abundance they have, and to see how they have changed from their initial soil type and to see whether these sterilised soils have actually held on to those microbes. But that's not necessarily realistic too much in a cropping system. So I've now got to find a way to apply those compositional changes to our cropping system. So what I'm going to do is another experiment. I'm going to take soil microbial communities from a range of different crop management systems. Not just that, but I'm going to take some from some estuaries, some sand dunes. I'll take some from some extreme volcanic soils. I'm going to create my own sort of mix, and I'm going to use some commercial inoculants. And then I'm going to put them into a sterile soil, and I'm going to grow a crop, probably going to be wheat. And I'm going to see if these changes in microbial composition or their community structure has a physiological effect on our ability to grow wheat. And so, in summary, I hope I haven't lost too many of you, and I hope I haven't confused you, but it's really understand that soil microecosystems are incredibly complex. They're forever changing, and they are very sensitive to environmental factors and variants. And so even if you were to sample on one day, in a week's time, if you come back and there's a change in the environment, you're probably going to get a very different microbial community than what you had the week before. And so going on from that, the presence or absence of specific species in your soil does not necessarily mean a loss or a gain of soil function. And I think the biggest take-home message is that a greater diversity in our soil microbes is our ability to not necessarily, I mean, farmers are always asking, will this increase my yield? And my answer is not necessarily, but it probably will protect you against greater drops in yield. So looking after our soil microbes isn't a increase in yield percentage per se, but it will stop fluctuation of yields. And that's probably what microbial diversity is good for. So thanks, everyone. Thanks for listening. Thanks,
Cameron. That's fantastic, and it's a really great, like you say, deep dive into the work that you're doing and the complexities of an environment that we're possibly trying to control, which we clearly can't, or we may be able to influence a little. So I'm happy to open the floor to some questions, because I'm sure there must be some questions from Cameron, because that was really interesting. And Natalie, do you have a question? I just noticed that you're unmuted.
The only query I had is, I'll admit, a little bit baffled, but that's okay. When you finish this work, are you sharing it? Like, how will you, I suppose, circulate that out? And are you making, I call them farmer-friendly versions? Because quite often PhD papers are not the easiest to read for the general paddock community.
Yeah, so I guess for now, I'm like any typical PhD student. I'm very excited about my research. I'm very passionate about it, and I tend to explode onto anyone who brings up that one topic that I'm interested in. But I do have a lot of experience. I was a techo at Wagga for 2.5 years running agronomy trials, so I do understand that information is key, but it's more delivering it. How you deliver it and what message you're getting across to growers is more important necessarily than what you're actually saying. So I think in terms of the messages and how we're going to get this to farmers, this is part of a soil CRC project. So my research will be taken up by the soil CRC, and then I'm assuming that it's going to be given to extension officers, or they'll probably get me to go out and talk. And obviously I'm going to have to change my message a little bit into, what farmers need to look at. But there are positions that are starting to be created now within the soil CRC, which is basically they're communicators, people who are good at talking. And so hopefully I'd love to get in on that and learn that skill and get that message to the growers.
Yeah, and I guess the beauty with the soil CRC is that they do have those connections with 19 grower groups, NRM organisations, to be able to, for want of a better word, ground truth or take it to the community and say, right, well, this is what we've learned. What else do we need to know? Or how can we then apply this, in the paddock? So. Yeah, and
the reason I'm not doing it right now is I don't want to go out to growers and go, we don't know. I mean, we're trying to find out, but we still don't know. So I'd rather wait until we actually have some solid data to be able to show them. And even if it's negative, I mean, we've already seen with one experiment that microbial inoculants aren't doing what they're saying they're doing, and that doesn't necessarily mean don't use them. It just means that the way that people think that they're working is not true. They're working in a completely different mechanism. So I think we're going to wait until we have some solid data before we start spreading the message.
Yeah, and we already have some knowledge around AMF, for example, where some crops are more likely to host vascular mycorrhizal fungi and other crops don't, and so that fungi decreases during that crop rotation. Is that right? Am I
correct? Yeah, no, that's and what we're finding. Yeah. Some people are trying to.
Yeah, go on.
Sorry, you go.
Do you think we'll get a rotation that you're saying you're going to use wheat? So do you think we'll get a rotation that says, well, if you grow these different crops in these sort of orders, it'll help flatten the ups and downs, or create a nice environment for each of those crops in turn?
Yeah, so I think what's happening is growers are already employing some of these techniques, and they're already noticing the end game. What they don't know is how it's happening and how to exploit it more so than what they already are. So I think that's where I come in, and that's where this research is going to help. Is the growers are already doing it. The intercropping, companion cropping sort of stuff, precision sowing, none of this is new to a lot of growers. That being said, they don't always get to the results that they're desired, and so they want to know why that's happening. And I suppose that's where we're going to come in. It isn't necessarily do these things that they've already been doing. It's we're trying to find out exactly the mechanisms that are involved, so that we don't make the same mistakes again.
Yeah, great. See you, Emma. Thanks for joining.
Bye, Emma. See you later.
Any other questions? A couple of minutes. Any questions? You've done a great job, Cameron and Justine. Really appreciate your time and effort in presenting to the group. Yeah, as you can see with the few questions we've had, it's a really sort of still unknown, and I guess that's why you're doing the work you're doing, Cameron, and why Justine's been working on it for many years. An unknown area, and trying to understand it and get a handle on where we're going. And I'm working with some grower groups, and they're interested in, they're doing different things like worm extract, taking worm castings and extracting biology from it and spraying it onto paddocks. There's things out there called Johnson-Su reactors, bioreactors, that they're using to create extracts from and spraying it out. And sometimes we're getting results from those, but we're not really understanding why, I suppose. So, I've got some grower groups that have been spraying biological material, and they are getting an increase in pasture production or an increase in dry matter production and these sort of things, but it's not really understanding why or being able to explain it, and whether we can repeat it as well. So we did it last year. It was really you know, great rainfall and great growing conditions and those sort of things, but we did get a difference between, you know, applied and not applied. But will we be able to repeat that in a dry season or in a, you know, particular?
And that's why most agronomy trials go over numbers of years, not just the one season. And a big problem with a lot of soil microbial research is a lot of it's done in growth chambers or, you know, glass houses. And then when they transfer those trials out into the field, they don't get the same results. And that's because there are so many more influential factors affecting microbial function in a field than you would find in, you know, in a glass house.
Yeah, great. Excellent. Okay, thanks Phoebe for your feedback. Yeah, if there aren't any other questions, I'll wrap it up. But I'm sure you know, Justine and Cameron would be happy to yeah, if you've got any questions that you'd like to ask offline, I'm sure they'd be happy to share or answer any of your questions. I think Justine, you gave me some links for some monitoring, some microscopes and those sort of things. So I'll share those again with this group. And yeah, if anyone's got any sort of monitoring questions of Justine, I'm sure she'd be happy to answer. And I'd be interested in what you guys are up to in regards to any biological monitoring that you might be doing, or if you have been monitoring, if you've been getting any results between different, if you've got particular treatments or whatever they might be. So feel free to share. So thanks again for your time, and just some housekeeping for the group this afternoon. We're catching up again with the wider community of practice. So the West and Northern groups and Southern groups are all coming together. And Hannah Beth Luke from Southern Cross Uni will be giving a presentation on decision making and working with landholders in regards to adoption of soil management practices. So she's been doing some survey work and has been doing a lot of research in that in recent times. And also Michael Roach from University of Tasmania is going to be talking about digital soil mapping. So he's been doing some work with UTAS in this environment that we're in, where we haven't been able to get out into soil pits, actually producing them online and presenting them in an online environment. So he'll run through how to do that, which will be really interesting. And hopefully we might be able to support the groups to do that, create an online soil pit or some digital soil mapping for each of the groups. So, yeah, we'll work towards that this afternoon, and that starts at 1:45 Australian Eastern Standard Daylight Savings Time. And yes, Phoebe, I'm recording this, so we'll be able to get a recording of this presentation, and I'll send you the link. And